surfaced. When that bay is still deep, the environmental sieve is essentially closed, save for a few aquatic species perhaps. So most of those seeds will either remain dormant or become senescent. As that basin fills in, however, flood tolerance or flooding stress decreases, so the somewhat flood tolerant species are able to pass through that sieve, begin to germinate, and contribute their own seeds back to the seed bank. And as that vegetation spreads, the relative contribution of local plants should increase compared to that of the river. So I use the principles of stratigraphy in order to investigate two hypotheses to determine how accurate my conceptual model is. First, I expected that before and after the formation of the marsh, we should see distinct plant communities or seed communities. Second of all, that as that vegetation develops and as we become more recent over time, which means closer and closer to the surface of the sediment, we should see greater similarity between the seed bank and the above ground vegetation. I accomplished this by sampling along the shorelines of the newly formed islands. First, in December 2014, I accessed 22 stations at which I took surface samples of sediment in order to collect seeds as well as assess the standing vegetation by percent cover. I returned in May of 2015, hit the same stations, again, assessed the percent cover. Additionally, at 10 of those stations, I took 10 centimeter deep cores in order to capture seeds buried in deeper layers. What I was looking for here was a signal of that 2011 flood. The dates in parentheses refer to when the samples were taken. The flood layer I was looking for should be described as very sandy, so large grain sizes, very mineral, very little organic content, and high density. After that marsh forms, you should see what I refer to as an emergent layer, which should have smaller grain sizes as those plants trap more fine grain sediments. Also, higher organic content due to contribution of biomass. And just for reference, when I refer to the surface layer, which I took back in December, that should be considered as a subset of that emergent layer, so it's not distinct, it's just the top 1 or 2 centimeters. And then, of course, we have our standing vegetation in 2014 and 2015. By analyzing a subsample of the sediment in each core slice, I measured grain size, organic content, and water content, and defined the boundary between the emergent and flood layer based on a jump in grain size and a decrease in organic and water content. It's worth noting, in one of the cores, I was not able to capture that flood layer, so I have 9 observations of the flood layer, 10 of the emergent layer. All the samples were treated with baking soda to deflocculate clay particles, which stick together, as well as peroxide to degrade some of the organic detritus that was making separation kind of a pain. I ran those samples through 2 sieves, air dried them, manually separated the seeds, and due to the semi-destructive nature of that treatment, the seeds could only be reliably identified to the genus level. To test for differences between the flood and emergent layer seed communities, I looked at a series of descriptions of biodiversity, richness, Shannon and Simpson levels. Those are all based on the same underlying mathematical equation, but they differ in their sensitivity to either rare or common species. Because I didn't have an a priori knowledge of what index I want to use, I utilized basically a simultaneous approach where you test all the different indices, correcting for multiplicity, which is multiple tests, adjusting your p-values essentially. And this was a bootstrapping approach implemented in the R package SimBoot. And as we'll see in a moment, I repeated that analysis, removing the most common genus from the data set. And to look at our local versus absent plants, I also ran a chi-square test on just the abundance of seeds either belonging to species present or absent in West Bay between the 2 layers. To assess similarity, I used what's known as Sorensen's Similarity Index, which is just based on presence and absence data, since above ground vegetation and seeds were measured on different units. It can generally be understood as the proportion of taxa in common between 2 samples. And because of the way this number distributes, I use non-parametric statistical tests. Taking a look at our two layers, we can see both of them are highly dominated by the seeds of Heliotropium, which is a cosmopolitan plant, but it has never been observed in West Bay by me. The seeds were highly degraded. As you can see on the right there, they were basically the remnants of the husk of the seed coat. Additionally, juncus was pretty prevalent in that flood layer. Again, also relatively degraded seeds. And Heliotropium decreased in abundance in the emergent layer. Running our diversity analysis in richness, there was no significant difference between the two layers. However, on both the Shannon and Simpson scales, the emergent layer was significantly more diverse than the flood layer. Taking Heliotropium out of that data set, all those significant differences disappear. That suggests, but it doesn't mathematically prove, that Heliotropium is mostly responsible for those differences. Moving on to our chi-square test, the emergent layer was significantly more represented by the seeds of genera that are present in West Bay. Those plants responsible for that shift include Amaranthus, Typha, Potamogeton, and Phragmites. Interestingly, Schonoplectus, which is highly represented above ground, actually decreased in abundance in the emergent seed bank. And once again, taking Heliotropium out of the data set, if we just look at present and absent genera, there's no significant difference. However, if we account for the proportion of Schonoplectus seeds, because there was such a decrease, it does come out as a significantly different distribution. Moving on to our above ground communities, at both points in time, they were mostly dominated by Schonoplectus, with a few other genera. As we can see, there's much lower richness above ground than below ground. That's not too surprising. And our surface layer generally resembled the emergent layer, although there was a higher percentage of Amaranthus that is an annual. It produces tons of seeds, so that could be just ineffective where I sampled when. Utilizing our paired non-parametric analogy to like a paired t-test, I found that the above ground vegetation became significantly more similar to the surface seed bank in 2015 compared to 2014. Additionally, comparing that above ground vegetation to the various layers, that above ground community in 2015 was significantly more similar to the surface layer than either the emergent layer as a whole or the flood layer. So I can conclude that both my hypotheses were supported. Definitely a distinct community between the two layers, and an increase in similarity as the seed bank gets more and more recent, suggesting that the local community is contributing to the seed bank. Those seeds are then contributing back to the local community. A few interesting takeaways. We showed that there's very likely a river signal associated with those two highly dominant but absent above ground seeds, Heliotropium and Juncus. Those could serve as a marker for river floods in other types of systems, not necessarily those two genera, but utilizing seeds that you can determine did not come from within the system, especially in cases where maybe there's not an obvious geological effect, such as grain size. We had these three taxa, Amaranthus, Typha, and Potamogeton, appear to be heavily contributing to the emergent seed bank after germination. They all show up within the first growing season. Interestingly, as I mentioned, Schonoplectus decreased in the seed bank despite being well represented above ground. What possibly could be happening here, Schonoplectus deltarum, which is one of the major Schonoplectus species out there, does germinate early on. It can germinate in slightly flooded conditions. However, in the first 2 or 3 growing seasons, it really invests in below ground biomass growth, clonal or vegetative, after which it sends up a lot of above ground biomass and forms either systems where it's dominant or even monoculture. So it could be happening as those original seeds were deposited during the flood, they begin to germinate, and Schonoplectus is not necessarily investing too much in reproduction by seed. I'm not going to talk about this too much. However, it's worth noting These two introduced genera, Phragmites and Alternanthera, were very well represented above ground throughout West Bay. That's common reed and alligator weed, respectively. Alternanthera has been shown to never produce seeds in its introduced range in other areas, such as Asia, and I did not find any Alternanthera seeds at all. I did find a few Phragmites seeds, especially near the surface. There's some disparity in conclusions about how much seeds Phragmites produce. There is a lot of genetic variation. However, these results suggest that these two genera might be decoupled from the process previously described. So moving on, now that some of these seeds are beginning to germinate, what happens when the adult plants grow up? How do they contribute to the geomorphology of the system? So it's understood that the above ground biomass of plants in wetlands produces friction that slows down the flow of water, promoting the deposition of finer grain sediments that would otherwise be washed out of the system. That promotes elevation gain. It also reduces wave energy that would cause erosion. While the below ground biomass takes up space within the sediment, causing, again, elevation gain. And it also physically binds its root hairs to individual sediment particles. That increases the cohesion of the sediment, which should, again, reduce erosion. Now, less erodible sediments associated with a river delta will exhibit this braided morphology to the left, where there are a lot of interconnected channels, a lot of channel switching, while more cohesive, less erodible sediments will display a channelized morphology such as that to the right. It's worth noting that before the evolution of vascular plants in the Devonian period, all river deltas found in the geologic record look like the morphology to the left. So plants have definitely been demonstrated to have a role in shaping the entire shape of the Earth. Moving back to our conceptual model here, we're now concerned with erosion. Again, as our plants begin to germinate, those roots should be decreasing erosion. Additionally, I would expect that those plants that have a heavy investment in below ground biomass should be better at holding that sediment together and therefore contribute more erosion resistant than more sparsely rooted plants. I tested these hypotheses again out in West Bay. I can't really measure erosion per se in this type of situation. However, you can measure shear strength, which describes the amount of force required to break sediment or soil. I accomplished this with basically a little vane you put in the soil. It has a torque meter on it. You can convert that into shear strength. And the two hypotheses: first, I needed to see a positive significant correlation between below ground biomass and shear strength. And if that was satisfied, I again expected to see that clonally growing plants such as grasses and sedges, along with woody species that have really large roots, should be associated with higher shear strengths than other types of plants or no plants at all. So I conducted two surveys in 2014. The first is part of a separate study. I hit 120 points throughout West Bay, regardless of whether they were new or old. At each station, I assessed percent cover of each species, as well as took a shear strength measurement at the surface. Returned in August of 2014, focusing on that new delta. I accessed 35 stations. At this point, I actually harvested biomass both above ground and below ground using a peat auger. I took a sediment subsample to assess water content, organic content, and grain size. And finally, again, took a shear strength measurement at the surface. Our stations fell into one of 5 categories based on vegetation. The first one being what I refer to as pre-emergent, which is anything not a marsh. That could be a mud flat, that could be open water, with or without aquatic vegetation. I had a forb community consisting of either Sagittaria, Typha, or Colocasia. I treated Phragmites as a separate community, again, because it forms these dense monospecific stands, and because if not invasive, it's at least cryptic for some of those haplotypes. Others are known to be invasive. So I kept them separate from the native sedges and grasses, including 2 species of Schonoplectus as well as Zizaniopsis. Finally, we had a woody community consisting of juvenile salix trees, as well as sesbania and amaranthus. To test my first hypothesis concerning root density and shear strength, I ran a linear regression on shear strength using multiple independent variables, below ground biomass being the one of interest, but I also included other geotechnical variables which have been shown in the literature to also affect shear strength, so keeping that variation separate. For the community effect, I just ran a simple analysis of variance on shear strength across community types along with a post hoc multiple comparison test, and because I selected sites based on different criteria in the two surveys, I ran separate analyses. So I did see a significant effect of below ground biomass on that shear strength. It did only account for 25% of the variation, but that was sort of to be expected because all these other physical factors also affect shear strength. Additionally, it had a significant negative effect of increasing water content on shear strength, especially at those small, muddy grain sizes. This is just real quick how those different community types were distributed throughout the bay as a whole, along with on those newly formed islands. I did find significant community effects on shear strength. In the bay-wide survey, I found that the native clonal sedges and grasses, along with the woody species, had higher shear strength in their sediment than any of the other three community types. In the smaller sample size August survey, I only found that those sedges and grasses were significantly higher in shear strength than the pre-emergent communities. However, my first hypothesis was definitely supported. We do see that positive association that suggests that the vegetation is not only decreasing the erosivity of the water through friction of its above ground biomass, but also should be reducing erodibility by its below ground biomass. This means that plants have the potential to affect the hydrology of a building delta, affecting the future geomorphology. That in turn affects where new plants can grow, so there's a potential feedback effect that definitely warrants further study. Additionally, we definitely found strong evidence for those native sedges and grasses, especially Schonoplectus, because it's so dominant out there, as one of the best plants for erosion control. As for the woody communities, we didn't see that significant effect in August. However, out of those 35 stations, I only had 2 that fell into the woody category, so that could just be a sample size effect. I can't really speak on that further. Interestingly, Phragmites I expected to be associated with higher shear strengths because it has such a dense below ground network. However, looking into the literature a little bit further, I found that Phragmites actually grows its roots significantly deeper in the sediment than other species. What that means for shear strength, you can have all the roots in the world, but because erosion takes place at the surface, if it's way down in the sediment, you're not doing anybody any good. So you might want more Schonoplectus deltarum and less Phragmites australis in those marshes that you're building. So I tested the interactions between those two species in the context of changing ratios of limiting factors, which was first described by Tillman. His original paper was based on a case study from Alaska. This was a primary successional environment where we had a really barren soil. Only one species of plant was able to grow and had very low nitrogen requirements. The rest of the system was nitrogen limited. However, as it grew, died, and returned to biomass to the soil, the nitrogen content of that soil begins to increase, eventually supporting a community of other species as their seeds are able to germinate. There's that environmental sieve. As they grow and form a canopy, the new limiting factor becomes light and the original species is eradicated. In our system, as that basin fills in, the initial stressor is going to be flooding in most cases. That's the original limiting factor. However, as elevation increases, flooding frequency decreases, and that means inundation is less and less of a limiting factor. And just from what I understand about river diversions, I came up with a few potential other factors that could replace flooding as possible limitations on the vegetation. First, As I mentioned, that Mississippi River drains over half the continental United States. A lot of that is agricultural land in the Midwest. There's a lot of fertilizer runoff. So these diversion basins could be subject to high levels of excess nitrogen loading. Second, as I mentioned earlier, as that first delta forms, it's very high in mineral content. That means it's very dense. It has low organic material. However, as plants begin to grow and contribute biomass into that soil, organic material increases, density decreases, and finally, the end member of a lot of these bays is the Gulf of Mexico. So especially during times of drought or low river flow, coupled with the inland southerly winds, these basins could be subject to saltwater intrusion events. And based on what I know about the life histories of the two species, this is Phragmites on the left and Schoenoplectus on the right, I came up with a series of hypotheses based on the following treatments. First, as nitrogen increases, because Phragmites, it's weird, it can do C3 and C4 photosynthesis, whereas Schoenoplectus is strictly C3. I expected that the more efficient user of nitrogen, Phragmites, would compete better against Schoenoplectus. Additionally, nitrogen loading has been shown to increase above ground biomass at the expense of below ground biomass. So I would predict a decrease in root to shoot ratios, which is important for shear strength. As salinity increases, although Schoenoplectus is one of the more salinity tolerant freshwater species, Phragmites is generally thought of as much more salt tolerant. So I would expect Phragmites to compete better under brackish conditions. Finally, as that soil organic content decreases and density increases, Phragmites is generally thought of as a more opportunistic species, quickly colonizes, again, these primary successional areas or recently disturbed areas, while Schoenoplectus kind of takes its time. Also, although soil microbial activity could be of interest, I didn't really look at that, but I did expect just a physical impediment effect on below ground biomass, meaning that the substrate is just harder to grow roots through. So I expected under that lower organic content, mostly sand, that root to shoot ratio again would decrease. I tested these treatment effects in a greenhouse experiment in 2015. I had 2 nitrogen treatments, 2 organic treatments, 2 salinity treatments, utilized a factorial block design, meaning I represented all treatment combinations in each of 3 blocks. This was also an additive pairwise experiment, meaning both species were grown both in competition and individual monocultures at densities consistent with field observations. I ran this experiment for 100 days, after which I counted all the stems of each species in each pot, measured total above ground, total below ground dry biomasses, got average measurements of stem height and diameter from 10 randomly selected stems from each pot, and assessed the proportion of stems that produced flowers by Schoenoplectus. Phragmites never produced any flowers during the experiment. And because a lot of these variables are correlated, I utilized principal component analysis to reduce the variables into fewer important scores, utilizing Varimax rotation in order to increase the independence between the different factors, and tested the treatment effects using a mixed model factorial analysis of variance on both those component scores and the root to shoot ratios, because again, we care about the effect on shear strength. So Schoenoplectus reduced down to 3 important components, one describing the productivity, one describing the reproductive potential, and one describing the average stem morphology. The big takeaway here is that Schoenoplectus did better under high organic content conditions. As you can see, much more productivity, higher root to shoot ratios with that high organic content under competition there. I actually saw an increase in root to shoot ratio when brackish conditions were introduced on the condition that LOI again was high. This possibly came at the expense of reproductive biomass allocation. So we see that under those brackish conditions, we see a decrease in that second principal component. As for Phragmites, there were only 2 components because there was no flowering, so we just had productivity and stem morphology. The big takeaway here is that the high nitrogen treatment, contrary to expectations, actually hurt Phragmites. So we see decreased productivity, decreased root to shoot ratio, even under the absence of all other stressors. So this is fresh water, no competition, high nitrogen, we got a lower root to shoot ratio. You may have noticed that all these treatment effects of the abiotic stressors were manifested in the monoculture treatments, while there was less difference in the competition effects. Additionally, those competition treatments consistently produced lower productivity scores than the monoculture treatments, meaning the competition was sort of had a greater effect on the plant biomass than any of those other stressors. This is not a totally novel finding, or it's not an unprecedented finding. Here's a study from 2001 where they tested 3 different wetland plants along 5 different levels of salinity, and he found that the basically the competition treatment reduced growth and productivity both independently of the treatment level and to a greater degree than the salinity treatment. In particular, this is one of my more interesting findings, that increase in root to shoot ratio. There's not a lot of literature on Shenoplectus deltarum. However, we can look at congeners. So this species, tricatter, has been shown to invest in building cortex and epidermis of its roots. This prevents water loss in brackish conditions, as well as increasing the aerenchyma, which is the airspace that conserves oxygen. Shenoplectus americanus, which is a very closely related species, has demonstrated that it reallocates biomass below ground under brackish conditions. And finally, Shenoplectus californicus. This speaks to why the lower density is important. Mark Hester recently published a paper where he showed that Shenoplectus californicus is limited in its below ground productivity by more dense soils. The nitrogen effect on Phragmites, I think, was just an unexpected effect of the way I designed my treatments. So I accomplished the nitrogen treatment by using this commercial fertilizer, which has a time release mechanism, and delivers the nitrogen in the form of urea, which is an organic nitrogen molecule. Generally, these fertilizers and their time release mechanisms exploit the nitrogen cycle by which the organic molecule has to be mineralized at least to ammonium, if not nitrified to nitrate before it's available to the plant. However, Phragmites, weird plant that it is, has been shown to be able to directly uptake organic nitrogen molecules, including urea. So what I think happened there was just the Phragmites got all that nitrogen at the same time and got, you over-fertilize a wall and it gets burned. Above ground biomass senesces. However, the literature does support the root to shoot ratio effect. Again, increased nitrogen will reduce the root to shoot ratio of Phragmites as well as most other species. Looking at this in the context of the strategy for operating these river diversions, as I showed earlier, the West Bay diversion was just an open, uncontrolled diversion. However, the planned ones for the future are going to have floodgates on them, so they're only open at particular times of the year. While they haven't totally settled on the exact strategy, they're generally talking about during winter periods of high water, having those gates open at all times, and then the spring flood, which is usually the highest discharge, opening the floodgates on the rising limb and then closing it at the peak in order to minimize shoaling downstream. In terms of what that means for my treatments, when those floodgates are closed, you're going to restrict the amount of nitrogen from upstream that gets introduced into the river. And then those basins could be subject, as I said, to those saltwater intrusion events, even if the river is not super low, because again, the gates are closed. So that would serve, if my results are able to be compared to natural systems, that would minimize the root to shoot effect on Phragmites caused by excess nitrogen, and possibly could take advantage of the root to shoot ratio increase of Shenoplectus. When you do have saltwater intrusion, that could possibly serve to actually strengthen the bush. contrary to what you might read on the comment section of NOAA.com, diversion articles. So just to sum everything up in my 3 chapters, first I demonstrated a difference in the composition of seed banks before and after the formation of a new deltaic marsh, described by the increase in similarity of the seed bank to locally growing plants. Second, I found a community-specific effect of plant below-ground biomass on soil shear strength. And finally, I found that in general, at least under the conditions that I tested, these 2 species are limited in their monocultures by abiotic stressors, but in mixture, they're limited by competition. And the theme that kind of ties all these chapters together is the relative shift in influence from allogenic factors, which exist outside the system, to autogenic factors, which describe those from within the system. So early on in that marsh, your propagules or seeds are going to be delivered entirely from the river, besides maybe a little bit of animal dispersal, birds flying over. The geomorphology is largely dictated by the hydrology of the river in the receiving basin and the sediment delivered. And finally, whatever vegetation does emerge is going to be limited by those abiotic stressors. As the marsh gets older, the propagules are increasingly going to be delivered from within the system. The geomorphology is now going to be modulated by the effect of vegetation, strengthening the soil, preventing erosion where it would otherwise take place. And finally, that as that vegetation gets more dense and begins to grow together, at least in some cases, it could be more limited by other vegetation rather than those abiotic factors. These all point to the role of vegetation as an important engineer of these wetland ecosystems. I feel that the work I've done definitely opens the door for a lot of future research. I'm going to describe just a few examples from my chapters. So I didn't really look at germination requirements of any of those seeds in my first chapter. They vary in their requirements for perhaps flooding or lack of flooding. Sometimes they require some type of physical abrasion. Sometimes they have to pass through the digestive system of an animal before they're able to germinate. And this is definitely known as a significant knowledge gap. This was excluded from master plan vegetation models for that reason, because of the lack of data. Practically, this could address, whether or not these big planting expeditions are really necessary in the building of wetlands. If the seeds are going to come in and sprout vegetation anyway, could those resources be better used elsewhere? Second of all, I only tested the shear strength effect at one point in time, really one growing season. It'd be interesting to look at perhaps how changes in vegetation community affect that shear strength effect. We do know eventually there's a second half of the delta cycle that I didn't talk about, where that land is eroded or sinks when those processes take over. So that could allow modelers to better predict the lifetime of these new marshes formed by the river diversions. That factors into cost-benefit analyses, etc. And finally, I only tested really one density of the 2 species in my competition experiment. It'd be interesting to look at perhaps what would happen earlier on in that Shonoplectus cycle, for example, where it just has a few, it's doing that below ground investment, but it hasn't become dominant yet. What happens when a Phragmites propagule finds its way into the middle? Would we still see that same kind of stalemate, or would we see competitive exclusion? And, all in all, I wonder if it'd be possible to engineer these systems to promote the type of plants that most help keep that marsh persistent, whether that's Shonoplectus, whether that's another species. It'd be interesting to figure that out. So I hope I've illustrated the importance of these interactions between ecological and geological factors, as well as the need for a lot more research pertaining to these interactions. So I'd like to acknowledge all the various funding sources. Came from a lot of different places at a lot of different times. Airboats are expensive. We wouldn't be able to do a lot of this work without it. I had a lot of support, definitely from my committee, Kaz, Alex, Mike, and Jordan. Again, I would not have been able to do a lot of this stuff without the people who volunteered to come out into the marsh with me, as well as my various airboat drivers, both from Lumcon and Deep Delta airboats. Those who don't fit in that category, Dave Corey from psychology helped me out a lot with stats. Kristen, or Andy Nyman and his postdoc Kristen at LSU helped me out a lot with seed chapter and the identification, which I would not be done with right now if not for them. Phil Pullman helped me out with some of the R analyses. This company, Polydrip, they're great. Out by Baton Rouge, if you ever need a watering system or irrigation system, call them. They'll set it up for you. And even before I really started a lot of this, Giannis over at UNO showed me how to use these shear vanes in the first place. I'd also like to thank Davey, Ryan, and Jack in our department for helping out with all sorts of logistical paperwork things, as well as Cindy and Chuck over at Lumcon, Shelly Moe upstairs at CBR. I forgot to write Marilyn from ENS. She also helped out a lot. And Trevor over at Wildlife and Fisheries helped put me up on Pass Lucia during a lot of these field trips. So that was great. And finally, I'd like to thank my family and all my friends and the other grad students in this department. Thank y'all for making this like not the isolated experience that I thought it was going to be. You know, so great. All right. 46 minutes, so it's fast.
Thank you. That was great. I thought that was fascinating. Are there any questions?
When you say that competition swamps out the Untitled Chapter, are you measuring your variables like biomass on per plant level or at a whole pot level?
At a whole pot level?
And was your design additive or was it substitute? Like, is it just because you might have had fewer plants in the pot compared to a full pot?
So it could have been that. I thought about that a lot. Like whether I should, in the monoculture treatments, cut them in half. But because I was actually expecting competitive exclusion, so I just did the half and half with the idea that maybe Phragmites would just overtake the whole pot, possibly, and ended up not doing that. I never saw any kind of exclusion. We did point out that the design of that, because it was in those constricted pots, they were really root bound. So, we talked about how Phragmites grows its roots deeper. It wasn't able to do that in those pots, so that could have been, a constraint on that effect.
You talked about the planting expeditions in light of the research that possibly, because all of these seeds and so on are positive, all this natural process going on. Now, my question is in relation to what you said about engineering marshlands. Can you just clarify, just in terms of what you learned from your research, what sort of approaches of engineering you think would be the most fruitful if you have some ideas about that?
Really, I didn't look at engineering at all. So there's certain things you can do in terms of diversion design. A lot of that's been done. So they've looked at, how deep to make that cut in the levee. So, making a really deep cut allows more sand from the bed of the river to get out into the receiving basin. If it's shallow, you might get some mud up on the top. That can still build some land, but again, it's not going to have that like high mineral component. It might be more erodible because again, those grain sizes are smaller and lighter. I didn't really test for I was originally going to test, instead of organic content, I was going to do a grain size treatment in that competition experiment. I had a hard time sourcing just plain silt that didn't already have a lot of nutrients or other organic contaminants in it. So just to simplify things, I went with density instead of grain size. But I I would suspect that maybe certain root hairs might be able to adhere better to larger grain size than smaller, but I can't do that.
Are there other questions?
Like,
Similar to the question I was just asked, so there's been a lot of money allocated to this since I was filling out planting. Right? And so one thing that I think about is it's even necessary to benefit others, rather than a process to see that these systems are not just for themselves, but also being vegetated markets.
I suspect that. If I had the resources or a future opportunity, I might go and access one of these planting sites and maybe look at the dynamics where planting has taken place and where it hasn't in a separate environment. I do think it's not necessarily in terms of growing a marsh. I think it has its place as sort of a publicity exercise. I'm not really kidding, but in terms of like relying on taking all these plugs out, that's heavy, that burns more boat gas. Relying on going out there and planting individual plugs of species that just grow anyway, I think that's definitely worth finding out before you spend all that money.
Are there other questions? Well, if there is one opportunity to ask more questions, I think we'll all be reconvening at the square at 5:30 today. We're a big fan. Funky Nation will be playing, so come if you have other questions.